Hello everyone, my name is Ashish from National Institute of Immunology, and today I'm gonna talk about Sestrin2, and I will talk about how Sestrin2 can facilitate glutamine-dependent cell survival and mitochondrial biogenesis in cancer cells under glucose-limited condition. Before starting my presentation, I would like to extend my thanks to the organizer for giving me this opportunity. And our lab works on deciphering the mechanisms behind cell death and cell survival. And while we talk about the cell survival and cell death, we should concentrate on the fact that we are talking about stress signaling. And at this stress signal, the which cell experience get integrates at the interface of the mitochondria. And it's mitochondrial response which decide whether cells will undergo cell death or cell survival. And two of the things, one is mitochondria, other one is metabolism. Both are very inextricably associated with each other. And while we are talking about these two terms, mitochondria and metabolism in concern with the cancer cells, then cancer cells have an ability to reprogram its metabolism in order to survive it under the harsh or adverse condition like nutrient deprivation or low pH condition or hypoxia, etc. So I would prefer to call it metabolic pleomorphism because of the fact that uh, cancer cell changing its metabolic phase from one to another in course of time with respect to availability of various nutrients in the environmental milieu. So there are, other than primary metabolites, there are other metabolites which we should target in order to facilitate or in order to get up to a proper cure about the cancer. So understanding various other metabolites like glutamate is very essential, and recent reports have shown that targeting glutamate metabolism, which can act as an alternative source of energy, can solve the function and can act as an Achilles heel as far as cancer cure is concerned. So when we talk about cancer, as we know, cancer reside in a very harsh or adverse condition where either nutrient deprivation is there or there is oxidative stress and other type of stress signaling. So there, one of the genes, one of the kind of genes, stress responsive genes are very essential to explore that what is the response or what is the way of regulation by which these stress responsive or stress inducible genes regulate various processes in the cells is very essential to decipher. One of such kind of family of genes which are evolutionarily conserved is cestrins. And earlier these proteins have been elucidated as the antioxidant protein, but in recent time it has been known that these proteins have a diverse role and they can modulate various other processes like autophagy, cell survival, mitochondrial health. And our recent reports have shown that Sestrin2 can facilitate mitophagy by helping pulpin translocation and under mild mitochondrial stress, while extensive mitochondrial stress can even degrade the Sestrin2 by making it more prone for ubiquitination in neuroblastoma cells. In this study, we are asking two questions. First one is how Sestrin2 can govern the glutamine metabolism under glucose-limited condition, and second is whether and how Sestrin2 can regulate the mitochondrial biogenesis under these low glucose conditions. To answer these questions, we cultured 8 different cancer cell lines in 3 different media. One is normal cell media where both glucose and glutamine was in optimal concentration. Other one is low glucose media where there was no glutamine, and then we supplemented the glutamine in low glucose media, and we found that in due to in low glucose media there was an increase in cell death after 48 hours, which was significantly rescued on glutamine supplementation in 6 cell lines out of 8, and we are showing here FG2 which showed maximum effect. And further to explore whether Sestrin2 has any role in governing this survival under glucose limited condition or not, we knocked down the cystrin under these conditions and then overexpressed the cystrin, and we found that knockdown of cystrin was able to increase the cell death under this glutamine high and glucose low condition, which was rescued on re-expression of cystrin2, by which looked for the colony forming potential of these cells. Then notably we found that specifically under high glutamine condition and low glucose condition, knockdown of cystrin was able to decline the colony-forming potential of cancer cells. On the other hand, surprisingly, we found that under normal conditions, a knockdown of cystrin was able to increase the colony-forming potential. This shows that the function of cystrin is very, very specific for as per availability of different metabolites. And in this case, under high glutamine and low glucose conditions, knockdown of cystrin can affect the colony forming potential in a negative way. As per as migration potential or invasion potential is concerned, we didn't get any change in any of the metabolic state. As per when we compare it with the siRNA, scrambled siRNA or knockdown of cystrin2. Then next question was, if cystrin under this high glutamine condition is affecting the survival, then can cystrin affect the glutamine metabolism or not? So if we look into the glutamine transport in the cell, then when glutamine enters into the cell, it gets converted into the glutamate at the interface of mitochondria, and then it gets converted to alpha ketoglutarate, which runs the Krebs cycle. So we knocked down the cystrin under this metabolic state, and we checked intercellular glutamine and glutamate level. We found a significant reduction in intercellular glutamate and glutamine levels under knockdown of cystrin, which was rescued on re-expression of cystrin2. So this decline could be due to two factors. One, less intake of or less transport of glutamine, and second, due to less of utilization of glutamine, and thus less of conversion from glutamine to glutamate. So we have used BPTS, that is GLS2 inhibitor, as a positive control, and we found that if you are using BPTS, then there was accumulation of intercellular glutamine. But this was not the case with cystrin2. We knocked down the cystrin2 and we found both intercellular glutamine and glutamate levels were less. So this shows that cystrin2 plays a role, or the decline in intercellular glutamine is most probably due to less of intracellular transport of glutamine. So how that affect the mitochondrial biogenesis, that is the second question. So one of the important marker of mitochondrial biogenesis is PGC1 alpha, and we evaluated that under different metabolic conditions like normal media, like high glucose and high glutamine media. And we found that under high glutamine and low glucose media, cells have more PGC1 alpha as compared to high glucose cells cultured in high glucose or low glutamine media. And both mRNA and protein levels were more as far as PGC1 alpha is concerned, but activation of cystrin was different. It was active in both of the cases. So to figure out whether effect of cystrin is specific, what is the effect of cystrin under glucose high or glutamine high condition, as far as levels of PGC1 alpha is concerned, we knocked down the cystrin under both of these conditions, and we found that decline in PGC1 alpha in response to cystrin knockdown was specific under these conditions where we have cultured the cell with high glutamine and low glucose media. To figure out further whether cystrin2 is specifically involved in the process or not, we use a triple knockout cell line where 3 isoforms of human cystrin 1, 2, and 3 were knockout, and specifically we reintroduce cystrin2 under glucose low condition, and we simultaneously increase the concentration of glutamine, and we found that there was increase in PGC1 alpha levels, specifically under the condition where we have re-expressed the cystrin2. It shows that cystrin2 is essential for regulating the PGC1 alpha levels under glutamine high and glucose low condition, and there could be other isoform involvement of other isoforms, but we have checked for cystrin1. Data is not here, but we didn't get much of the change. This increase or decrease, mainly if I will talk about the cystrin2 downregulation and low levels of PGC1 alpha, then this decline could be due to post-translational modification like ubiquitination, the degradation of the PGC1 alpha, and earlier reports also have shown that under low glucose condition, PGC1 alpha undergoes ubiquitination. So in our case, when we culture the cell under glucose high and glutamine high and glucose low condition and use proteasomal inhibition, we didn't get any of rescue in the level of PGC1 alpha. When we evaluated the mRNA level in triple knockout cell versus where we have reintroduced the Sestrin2, we found that mRNA level was less in a triple knockout cell, which was rescued on re-expression of Sestrin2. While we check for the mitochondrial biogenesis by evaluating different mitochondria encoded genes and also by analyzing the mitochondrial green intensity, then we found that knockdown of cystine was able to decline the mitochondrial biogenesis or my total mitochondrial content. And as per, if we parallelly, it is essential to look at whether the mitochondria is active. What is the role of cystine2 in governing the activity of mitochondria? So we knocked down the Sestrin2 and we checked the ATP forming potential of these cells under this specific metabolic condition. So we found that ATP forming potential was significantly declined in cystine knockdown. The next question was, as we have found that knockdown of cystine resulted into less of glutamine and glutamate levels. So if and we speculated it could be due to less of intracellular transport of glutamine. So thus we checked for glutamine transporters ACT2 and LAT1 in response to different metabolic conditions, and specifically we were finding positive results in concern with Sestrin2, and we found that glutamine high and glucose low conditions. Under these conditions, if we are knocking down the Sestrin2, then the levels of ACT2 and LAT1, these two transporters of glutamine, were less as compared to wild type. So it shows that Sestrin2 down regulation resulted into less of expression of these two, ACT2 and LAT1, two of glutamine major transporters. And to confirm these results, we reintroduce the Sestrin2 under Sestrin knockdown condition, and we confirm the results that under these conditions, Sestrin2 positively influence the expression of glutamine transporter LAT1 and LAT1 and ACT2. There is one question. I will take it lately. Thank you. So the next question was, till now we have explained that Sestrin2 is involved in glutamine transport, and that positively under these conditions where high glutamine is there and less glucose is there, it affects PGC1 alpha or mitochondrial biogenesis in positive manner by most probably by affecting the intracellular transport of glutamine. The next was how Sestrin2 can do that. So we analyze PGC1 alpha can be regulated by at transcriptionally can be regulated by different transcriptional factors, and few of them are these, and we analyzed the cytoplasmic versus nuclear transport, and we checked for nuclear transport in response to Sestrin2, and we found that triple knockout only FOXO1 was able to under the triple knockout of Sestrin, the translocation of FOXO in nucleus was significantly inhibited, and only under the Sestrin2 re-expression, specifically under expression of Sestrin2, this FOXO1 was able to restore. So this shows that FOXO1 could be the factor which is regulating the expression of PGC1 alpha in this case. To further dissect the upstream signaling, we use different pharmacological inhibitors of different kinases like ERK, GNK, P38, AKT, which influences FOXO1 and PGC1 alpha. And reports have shown that, and we found that only specifically the inhibition of GNK1 resulted in less of translocation of FOXO. So it means that for Sestrin2 is influencing the FOXO1 translocation through GNK1, and earlier studies also have shown that Sestrin2 can regulate the GNK activity. So, and we also checked the mRNA level of PGC1 alpha under these conditions where we suppressed the JNK levels, and we found that there were less of PGC1 alpha levels under JNK inhibition or Sestrin2 knockdown, and re-expression of Sestrin2 in knockdown cells can increase the levels of PGC1 alpha. But interestingly, we found if we reintroduced the Sestrin2 but inhibited the JNK, then this increase was not there. So it means that JNK activity is also essential for governing the trans activation of PGC1 alpha. Further, we found that how Sestrin2 can further we a question that how Sestrin2 can influence both JNK and FOXO1. So we found that Sestrin2 inhibition can increase the activity or phosphorylation of status of JNK1, that is hyperactivation. We believe that it is hyperactivation of JNK1, and we found that specifically under these glutamine high and glucose low conditions, Sestrin to form a complex with JNK1 and FOXO1, and this complex was inhibited when we have triple knockout, and triple knockout where no cystine is there. While reintroduction of Sestrin2 specifically was able to restore this interaction of JNK with FOXO1. So it means JNK is forming, Sestrin2 is forming a complex with JNK and FOXO1, and maybe Sestrin2 is acting as a scaffold protein which modulates the JNK activity and influence the interaction between these two molecules, which thereby influence the nuclear translocation of FOXO1. So, as we have shown that earlier, I had shown that if you are knocking down the Sestrin2, then there will be less of PGC1 alpha. But if you are reintroducing the Sestrin2 and inhibiting the JNK, then also there will be less of PGC1 alpha. And while we check the translocation of FOXO1 in this case, was less of FOXO1 alpha as well in nuclear fraction. So it shows that activity of JNK is equally essential to influence the nuclear translocation of FOXO1. Well, on the other hand, while we checked the viability of cells under these conditions, we found that inhibition of JNK was able to rescue the. There was a marginal difference in cell death, but as we have found this result earlier also, that downregulation of Sestrin2 resulted into more of cell death. While if you suppress the JNK in this case, where we have less of Sestrin, we were able to rescue the cell death. So it means that, previously I have shown that Sestrin knockdown resulted into more of JNK activity. So it could be due to the, it could be possible that JNK, Sestrin2 is affecting and restricting the pro-apoptotic trace or pro-apoptotic axis of JNK. So with this, I would like to conclude my results that under glucose starvation, when glutamine comes in under glucose starved condition, Sestrin gets activated, which influence LAT1 and ASCT2 by an unknown pathway, and Sestrin2 activation can result in survival of cancer cell, and Sestrin2 activation under these conditions forms a complex with JNK and FOXO1, thus resulting into nuclear translocation of FOXO, which transactivates PGC1 alpha and which results into more of biogenesis of mitochondria. So, thank you very much. And these are all our in vitro studies. Right now, we are shifting our axis, and we are trying to evaluate that what is the role of Sestrin in development of cancer cells or liver cancer cells in vivo using in vivo models and patient sample. We are trying to speculate what is the role of Sestrin2 as far as development of liver cancer is concerned. Thank you very much. I will take some questions. First question is why metabolism. So, as far as glycolysis is concerned, recent studies have shown that because while we talk about glycolysis, we used to take glucose in consideration, but recent studies have shown apart from glucose, there are other metabolites like glutamine or even fatty acid. They can turn the oxidative phosphorylation on, and that can for the keyboard resistance. So I would suggest that it was earlier notion that glucose is essential, or glycolytic metabolism is essential. Now, if you are talking about chemo resistance, that I would say, or cancer cell survival, then I would say there are other metabolites which are essential to consider. Second question is,
Thank you.
do
Thank you,
different
Ashish.
cell types?
Just for the interest of time, we'll invite you to answer that question directly in the chat box. Okay?
Okay, okay, fine, fine. Thanks. Thanks a lot.
Okay, great. Not a
Yeah.